My name is Helena Yu. I am an associate attending in medical oncology at Memorial Sloan Kettering Cancer Center in New York City. I'd like to welcome you to today's activity entitled Understanding the Unique Role of HER3 in Oncogenesis. So what we will discuss today, we will have a brief introduction to the HER family of genes. We'll review the oncogenic role of HER3, go over molecular testing in non-small cell lung cancer. Where does HER3 testing fit in? We'll go over HER3 targeted therapies currently in clinical development, then end on the clinical relevance of NRG1 gene fusions and the use or investigation of HER3 targeted agents in NRG1 fusion positive cancers. So first, a little background on the HER family. So the HER family of genes encodes a highly homologous transmembrane tyrosine kinase receptor family. These proteins are broadly expressed on numerous cell types within the body, including epithelial and mesenchymal cells. The different HER proteins have similar structure with an extracellular, transmembrane, and intracellular component. These proteins are prompted to both homodimerize as well as heterodimerize, and this dimerization triggers kinase activity. There is overlapping downstream signaling pathways, including PI3K and MAPK, and activation of these proteins and downstream signaling regulates key cellular processes, including proliferation, differentiation, and survival. HER3, as one of the HER family proteins, is a unique receptor. It actually has limited intrinsic kinase activity. It signals exclusively through heterodimerization, so no homodimerization with other HER family members. HER3's preferred partners are actually HER2 and EGFR. The protein HER3 is activated by ligand binding. The HER3 ligands are neuregulins, otherwise known as heregulins, and these are encoded by 4 genes, NRG1 through 4, which will be relevant later. HER3 acts as a signal amplifier. HER3 contains multiple PI3K binding sites and potently activates this downstream signaling pathway. HER3 overexpression is observed across numerous types of cancer, including breast, lung, and colorectal cancer. We actually see gain-of-function HER3 genomic alterations rarely, but they do occur. HER3 overexpression in and of itself is not oncogenic, meaning it does not cause cancer, although it does promote the oncogenic activity of other HER receptors, particularly HER2. So the HER2, HER3 heterodimers actually possess the highest oncogenic capability of all HER family dimers, and this heterodimer of HER2, HER3 potently activates PI3K and MAPK signaling in a ligand-independent manner. And this signaling promotes cancer cell proliferation as well as survival. HER3 overexpression is associated with poor prognosis in several tumor types and is linked to resistance to several kinds of anti-cancer therapy, such as HER2-targeted therapy and endocrine therapy in breast cancer. HER3 is commonly expressed in the vast majority of primary lung cancers. There is actually correlation between the HER3 protein overexpression and either HER2 protein overexpression or the EGFR mutation, which we commonly see in non-small cell lung cancer. About a quarter of non-small cell lung cancers demonstrate the highest level of HER3 expression, IHC score 3+, which we'll discuss later. HER3 expression is associated with decreased time to metastatic progression and shorter time of relapse-free survival. There is a possible role of HER3 in resistance to EGFR TKI therapy in EGFR mutant lung cancer. In gefitinib-resistant cells, oncogenic MET protein phosphorylates HER3, which leads to activation of the PI3K pathway. And interestingly, HER3 expression is strongly upregulated in osimertinib-resistant cancer cells compared to osimertinib-naive cancer cells. I think these figures really here illustrate 2 things. The figure on the left illustrates the frequency of HER3 membrane staining by IHC in non-small cell lung cancers, and you can see by the rows surrounded by red that the majority, about half of Non-small cell lung cancers have high HER3 expression, indicated by IHC 2 plus or 3 plus. And then the figure on the right really illustrates that there is correlation or overlap between HER3 protein expression and other oncogenic drivers. So you can see that there is overlap between HER3 overexpression and EGFR mutant lung cancer, as well as HER3 overexpression and HER2 amplification or overexpression, which is a known established driver in lung cancer as well. HER3 is expressed in the majority of breast cancers as well, and its expression does vary to some degree with breast cancer subtype. Co-expression with HER2 is common. The vast majority of HER2 amplified breast cancers also demonstrate HER3 membrane staining, but we also see HER3 expression in breast cancers without HER2 positivity. HER3 expression is more common in metastatic breast cancer samples, and it's correlated with poor prognosis, which is also dependent on subtype, and it's also correlated with resistance to hormonal therapies, including tamoxifen and fulvestrant. We also see high levels of HER3 expression in the majority of colorectal cancers. And interestingly, the expression is higher in lymph node metastases compared to primary colorectal cancers. In colorectal cancer as well, HER3 expression is associated with worse clinical outcomes, specifically overall survival and disease-free survival. And high circulating levels of heregulin, which is the HER3 ligand, confers resistance to cetuximab, which is an EGFR antibody that is commonly used as treatment in colorectal cancer. The figure on the bottom of the slide illustrates HER3 expression in colorectal cancer, and you can see that there is a shift to the right where more primary tumors have HER3 expression, and a further shift to the right towards high expression in the lymph node metastases, suggesting there is a role in metastatic spread. Non-small cell lung cancer is a prime example of precision oncology. And by that, we mean selecting a patient population with a known molecular target to optimize clinical benefit from a targeted therapy. The figure on the left is the mutation pie that is often seen when describing non-small cell lung cancer. National guidelines recommend broad molecular profiling for these different mutations via a multiplex platform such as next-generation sequencing. All patients' cancers should be assessed for actionable driver alterations in EGFR, KRAS, ALK, ROS1, BRAF, NTRK, and RET, because these targeted genes have associated targeted therapies. And of course, there are FDA-approved companion diagnostics that assist us in this molecular profiling, but most commonly, we use a multi-gene panel using next-generation sequencing. So here is an excerpt from the NCCN guidelines. So if a patient presents with advanced or metastatic non-small cell lung cancer, it's first important to establish histologic subtype, specifically non-small cell lung cancer versus small cell lung cancer, and the different non-small cell lung cancer subtypes. We'll focus on adenocarcinoma. For patients that are diagnosed with adenocarcinoma of the lung, again, EGFR, ALK, KRAS, ROS1, BRAF, NTRK, MET-exon 14, and RET should all be assessed. And this should be assessed prior to initiation of first-line therapy because many of these treatments are targeted therapies that are approved in the first-line setting. And then also, of course, for all patients, we should test their lung cancers for PD-L1 expression because this, again, helps dictate the appropriate first-line therapy for our patients. So protein expression levels can also serve as biomarkers to select patients for targeted therapy, and this is in addition to the genomic biomarkers that we just said should be done for all patients. So there are already FDA-approved protein expression biomarkers that are companion diagnostics for approved FDA treatments. The best examples are assessment of HER2 expression in breast cancer, and this is to select patients who would benefit from HER2-targeted monoclonal antibodies or antibody-drug conjugates. We look for PD-L1 expression in many different cancers, and this selects for patients that may have especially increased benefit with immune checkpoint inhibitors or immunotherapy. And these protein expression biomarkers can be predictive, as just discussed, but they can also be prognostic biomarkers. An example of this is Ki-67, which is a prognostic biomarker in breast cancer, lung cancer, and other cancers. There are also investigational protein expression biomarkers, and these include TROP2, Nectin-4, MET, HER3, and heregulin. These are all investigational protein expression biomarkers because there are investigational targeted therapies, specifically antibodies and antibody drug conjugates targeting these proteins that are in clinical development. And then I think appropriate to highlight again, so protein overexpression and driver mutations are not mutually exclusive oncogenic drivers. So a patient can have an EGFR mutant lung cancer, but also have that cancer express HER3, which later might suggest responsiveness to a HER3-directed therapy. I think it's useful to spend one moment talking about IHC and H scores to help us better understand the scoring of protein expression. So immunohistochemistry, IHC as for short, is used for assessing protein expression levels. Antibodies targeting the protein biomarker of interest linked to fluorescent dye allows for visualization. You can do IHC staining on fresh or frozen tissue, and then samples are determined to be negative or positive based on a specific threshold. So IHC is more of a qualitative assay, and you can see that the figure on the right shows HER3 IHC staining in non-small cell lung cancer, and you can see an IHC score of 0 plus really shows no staining, 1 plus, 2 plus, 3 plus, you can see that there is increased intensity of staining as well as more diffuse staining. A more semi-quantitative method for looking at protein expression can be used when we assign an H score. This is when we determine membrane staining intensity for each cell over a given fixed visual field. And then we apply a formula to this membrane staining intensity to give a score ranging from 0 to 300. And there's more weight given to higher intensity staining to give a more quantitative picture of expression. Currently, there are no established biomarkers available or in use for HER3 targeted therapy. There are several biomarkers that are being explored. One, of course, is degree of HER3 expression. Also, there is some studies that are looking at heregulin expression. Remember, heregulin is the ligand for HER3. Their high heregulin levels are also seen in a significant minority of solid tumors. And again, this might identify patients with better response to HER3 targeted therapy. All the results to date have been quite mixed. Another possible biomarker is expression of HER3 negative regulators, specifically the E3 ubiquitin ligases. I think a problem here with assessing for biomarkers is the lack of standardized measurements to define low and high expression levels, which really plagues protein expression as a biomarker. And I think one way to potentially get around this is, are there specific genotypes or patient subsets that might be enriched for these biomarkers? And one such example is EGFR mutant non-small cell lung cancer, where most, if not all, EGFR mutant lung cancers have some degree of HER3 expression. The figure on the right illustrates progression-free survival in heregulin high and heregulin low tumors following treatment with a HER3 ADC. You can see that there might be some trend of longer PFS for patients that have high heregulin expression, but no clear answers quite yet. So that leads us to discussing HER3 targeted therapy currently in clinical development. The first in class in regards to antibody drug conjugates is a drug called patritumab deruxtecan. Another name for it is HER3-DXd. So antibody drug conjugates are multi-component agents. They consist of an antibody. So for patritumab deruxtecan, patritumab is an anti-HER3 antibody that is linked with a linker to a payload. And the payload in this instance is DXd, which is a DNA topoisomerase inhibitor. The way that these agents work, these ADCs, in particular pat-deruxtecan, the fully human IgG1 HER3 antibody patritumab binds to HER3 on the tumor cell. Remember, HER3 is preferentially expressed on these tumor cells, on the tumor cell surface through its extracellular domain. This binding leads to rapid internalization by endosomes. The endosomes, once intracellular, fuse with lysosomes, and the lysosomal enzymes actually break the linker between patritumab and the payload. And then the topoisomerase inhibitor payload is released. And then that chemotherapy payload blocks DNA replication, stimulates DNA damage, and triggers apoptotic cell death. So basically, the antibody portion of the ADC brings the agent to the appropriate cancer cells. And then once internalized, the payload is what is cytotoxic and deadly to the cancer cells. So patritumab deruxtecan in lung cancer was first studied in EGFR mutant non-small cell lung cancer. It was studied in a global, open-label, multi-center phase 1 study conducted at 22 sites globally for patients with metastatic EGFR mutant non-small cell lung cancer. The study did allow stable brain metastases, and the study did not select patients based on HER3 expression, but did mandate pre-treatment tumor tissue for retrospective analysis of HER3 expression done later. So the dose escalation portion of the study assessed escalating doses of HER3-DXd, and the primary cohort of interest was cohort 1, which was, again, patients with EGFR mutant lung cancer after prior EGFR TKI therapy, as well as after prior platinum-based doublet chemotherapy. Here are the demographics of patients treated on the study. The study population was relatively young, with a median age of 55. There were more females than males. Performance status was good in most of the patients enrolled. Again, patients with brain metastases were allowed, and about half of patients actually had a history of CNS metastases. The study did allow different EGFR sensitizing activating mutations. The majority of patients either had exon 19 deletions or L858R, but some rare EGFR mutations were allowed. The study population was heavily pretreated. The median prior lines of systemic therapy prior to study enrollment was 4. And as part of the eligibility, all patients had received prior EGFR TKI therapy. The majority actually received prior osimertinib in particular, and the majority also had platinum-based chemotherapy. And about half, 40% of patients, had prior immunotherapy as well. So here's the efficacy data in that phase 1 study. The overall response rate was 39%, with a median progression-free survival of 8.2 months. You can see on the bottom of the slide the waterfall plot that shows the percent shrinkage of patients on the study. What I think is important to highlight is that responses were observed in patients with a broad range of EGFR TKI resistance mechanisms. So patients that had on-target resistance, such as acquired EGFR alterations, as well as patients with off-target resistance mechanisms, including MET amplification, BRAF fusions, were all seen to have responses to this medication. And another important point is that efficacy and responses were also observed across a broad range of HER3 expression levels. So efficacy and response to the drug did not clearly track with HER3 expression. Pat-deruxtecan was generally tolerable with a manageable safety profile. Grade 3 treatment emergent adverse events did occur in most patients. The most common grade 3 treatment emergent toxicities included thrombocytopenia, neutropenia, and fatigue. They were generally early in the treatment course, and some or many were transient, and there was a relatively low rate of treatment discontinuation due to adverse events. The table on the right shows treatment emergent adverse events that were at a frequency of 20% or more on the study, and you can see the common side effects that were seen include fatigue, nausea, thrombocytopenia, anorexia, vomiting, and alopecia. Remember, these ADCs are chemotherapy, so they do have some of the more common side effects of chemotherapy, such as cytopenias, alopecia, nausea. So patritumab deruxtecan was also studied in metastatic breast cancer. There was a dose escalation portion where patients were given escalating doses of HER3-DXd. There were several patient populations of interest. The breast cancer study actually looked at HER3 expression prior to enrollment and enrolled patients that had high HER3 expression that were HER2 negative with metastatic breast cancer. They also looked at low HER3 expression and as well as HER2 negative metastatic breast cancers, and then looked at HER3 high triple negative breast cancers. And then you can see in terms of efficacy, it was actually quite varied. The study populations were different and small, but response rates varied from 10% to 30%. The agent was well tolerated. Few patients discontinued treatment due to adverse events, and the most common treatment emergent adverse events were similar to the lung cancer population: neutropenia, thrombocytopenia, leukopenia, and anemia. I think other things to note is this was median age of 56 in terms of the study population, good performance status, but this was a very heavily pretreated study population with a median number of prior therapies of 6. So in terms of ongoing clinical development of HER3-DXd, there is a phase 2 study in EGFR mutant lung cancer with the same eligibility as the phase 1, where patients were eligible after treatment with osimertinib and after treatment with chemotherapy. There also is a combination study of HER3-DXd with osimertinib, and this is in EGFR mutant lung cancers after progression on first-line osimertinib. And then there are other studies looking at HER3-DXd and breast cancer, including one that's looking at metastatic breast cancers that were previously treated and are hormone receptor positive, metastatic breast cancers that are HER2 positive but TKI naive, and then treatment naive early stage breast cancer. So in different stages and different populations, this drug is being looked at in breast cancer. And so to switch gears a little bit and to talk about NRG1 fusions, so fusions involving the NRG1 gene, which remember is the gene that encodes the HER3 ligand, were identified in lung cancer in 2014. Chromosomal rearrangements here result in part of the NRG1 gene being fused to one of several genes, most commonly CD74. NRG1 fusions are, after being identified in lung cancer, were subsequently identified in a small percentage of patients across tumor types. The figure on the right shows the rate of NRG fusions by tumor type, and you can see there is pancreatic ductal adenocarcinoma, renal cell carcinoma, ovarian, lung cancer, very small percentages, but is seen in a wide array of different solid tumors. And there's growing body of evidence that these fusions are oncogenic and potentially therapeutically targetable. Naked HER3 monoclonal antibodies have previously been tested and really have not shown efficacy in broad unselected populations, but these are now being looked at with renewed interest, specifically in NRG1 fusion positive cancers. So one of these such antibodies is seribantumab. So seribantumab is a fully human IgG2 anti-HER3 monoclonal antibody. It blocks ligand-dependent activation of HER3, HER3 dimerization with HER2, and other HER family members. And in preclinical models harboring NRG1 fusions, seribantumab reduces phosphorylation of HER family receptors and block downstream signaling. There was a phase 2 clinical trial in non-small cell lung cancer, ovarian, and breast cancer, and there did seem to be potential clinical benefit, not in all comers, but in a subset of patients with HER2-expressing tumors. And there is the phase 2 Sherlock study that evaluated seribantumab and docetaxel in patients with previously treated HER2-expressing non-small cell lung cancer. But this study actually was a negative study. It failed to improve progression-free survival compared to docetaxel alone and was terminated early. So I think this illustrates that heregulin expression does not seem to be the appropriate biomarker for these naked HER3 antibodies, but perhaps NRG1 fusions may be the right biomarker. So to test that hypothesis, the phase 2 Crestone study is currently ongoing, and that is looking at seribantumab in tumors, solid tumors with NRG1 fusions. And this is ongoing open label phase 2 tumor agnostic basket trial of seribantumab. And of course, patients need to have metastatic, treatment refractory, NRG1 fusion positive solid tumors. And the primary endpoint of the study is objective response rate. There are different cohorts that are being assessed in this Crestone study. The pivotal cohort are patients that have not received prior HER3 targeted therapy, and these are patients whose NRG1 fusion status was able to be centrally confirmed by NGS. And then there are small exploratory cohorts, including a small cohort of patients that have already progressed on HER3 targeting therapy, and then an exploratory cohort that has patients that have NRG1 fusions identified locally that weren't able to be centrally confirmed. And this study is currently ongoing. Another HER3 antibody is one called zenocutuzumab, and this is a bispecific IgG1 antibody that simultaneously binds to HER2 and HER3. It has a dual dock and block mechanism of action. It blocks interaction between both HER3 and the ligand heregulin, as well as between HER3 and HER2. And these interactions prevent downstream signaling through the PI3K-AKT pathway. In preclinical models, zenocutuzumab inhibited growth of NRG1 fusion positive cell lines and xenograft models. And this preclinical efficacy led to orphan drug and fast track designation, specifically for the treatment of pancreatic ductal adenocarcinoma. So zenocutuzumab is also being assessed in NRG1 fusion positive solid tumors. There is an open label multi-center phase 1, phase 2 study that is looking at this in metastatic refractory to treatment solid tumors that are NRG1 fusion positive. And this medication is given intravenously every 2 weeks. So there have already been a decent number of patients enrolled, and zenocutuzumab does appear to be active across multiple NRG1 fusion positive solid tumors. You can see from the waterfall on the right the percent shrinkage of different patients enrolled, and you can see that patients with non-small cell lung cancer in light blue and pancreatic adenocarcinoma in orange do seem to derive efficacy from zenocutuzumab. And this population also was highly pretreated, and responses were rapid and durable. So we wait to see additional data on this agent. In regards to safety, this drug appears to be remarkably well tolerated. When we assess treatment-related adverse events that were present in 10% or more of patients, it really was only fatigue, diarrhea, and nausea. So it does appear to be a very well-tolerated drug. So in conclusion, HER3 is overexpressed across a range of different cancer types and boosts the oncogenic activity of other HER receptors. HER3 overexpression is linked to poor prognosis and resistance to numerous types of anti-cancer therapy. One HER3 targeted antibody drug conjugate, patritumab deruxtecan, has demonstrated promising activity and safety in patients with EGFR mutant, EGFR TKI resistant non-small cell lung cancer, as well as metastatic breast cancer, and additional confirmatory studies are ongoing. While HER3 targeted monoclonal antibodies have had limited success in unselected patient populations in clinical trials to date, current efforts really focus on patients with NRG1 fusion positive cancers. And in that select, albeit rare patient population, there does seem to be efficacy. An appropriate biomarker for HER3-directed treatment has not yet been identified. Ongoing studies are focusing on specific disease types or disease subsets, such as EGFR mutant lung cancer, or driver mutations, such as NRG1 fusion positive cancers, to help select patients most likely to benefit. And there's ongoing research and work looking at HER3 expression or other protein expression biomarkers to see if they would be useful for stratifying patients that benefit from these drugs. I want to thank you all for your participation in today's activity. For those of you interested in claiming CME credits, you'll now need to complete a short activity post-test and evaluation to get CME credit. Thanks for listening.